So it's really, really nice to have so many of you here. I'm going to share a few more, like a little bit more info about how to stay up to date on the schedule, because it's iterating between AM and PM, and sometimes not not regularly. So I will share a little bit more info about how to join those salons in the chat, but I really want to take as little time away as possible from introducing Andre. Andre Watson, I have first met you actually at Burning Man. We were in the same camp together, and little did I know. I think it's always really interesting when you meet people in a social setting, kind of like the craziness that you find out about them. Once you're aware of what they do professionally. It took me a little bit to figure that out, and I think since then, your Ligandal is mentioned. Really, I think. Yeah, quite regularly. I think, especially in relation to nanotechnology, which is one of Foresight's main focus areas. So we're really happy to have you here. I think Ligandal's mission is important in a time of not COVID. Now I think you have something particularly interesting to share, which you already gave us a sneak peek into at a previous longevity investment salon that we did. And now I can't wait for what you have to show us. And I will post your bio, and I will post the description of the salon in the chat here. And with that being said, I want to give it up to you. And yeah, take it away, Andre. Show us what you got.
All right. Well, to start, this green thing here is the coronavirus spike protein. So if you imagine the virus having many of these spikes basically arranged like in a sphere around this larger virus, this is like one of those spikes. So, the question is, how do you create vaccines? How do you create therapies or antidotes against coronavirus? So one of the things is that COVID, or SARS-CoV-2, binds to is the ACE2 receptor, which is this red thing here. So this is a receptor. It's found on the cell membrane, and it also floats around in the blood. And this guy gets bound to by the coronavirus spike protein. And then the receptor basically gets pulled into the cell, and that's how the virus enters. So when we talk about developing drugs, one of the ways you can develop drugs against this is to create these sort of binders that will inhibit the interaction between these two things. So what we did is we actually built these binders based on, let me hide that. Right. So we build these binders based on essentially extracting a smaller portion of the spike protein and then actually looking at just the binding interface. And what you see here in green, the green parts are the actual amino acids that are sticky. So these stretches here. And basically I predicted these amino acids being the two stretches that are important for binding back in like early February. And then the question was, can you create a mimetic sort of structure? So can you take some smaller structure here that reproduces that binding pocket? And then secondly, can you further modify that to have sort of these other amino epitopes or regions that your immune cells can bind to and generate an immune response? So when we look at the SARS spike protein and its whole region here, zooming out, there are several parts that are actually immunogenic. So when you look at something like Moderna's vaccine or some of the vaccine approaches, people are actually printing out this whole big protein, right? And like these pink parts or these light pink parts are actually two of the regions that your T cells will generate immune responses to, whereas separately, your body will generate immune responses against some of these other stretches. So then the question was, how do you create sort of these peptides? Because they're pretty large for synthetic peptides. We wanted to be able to basically print them out and then have, these guys looking like things that you could basically create. So the next step was using a neural network deep learning simulation approach, actually figuring out what these guys look like, because they're not going to look the same as short versions of themselves as they do when they're part of that whole spike protein. So basically, we got many of these different potential structures elucidated from this, and basically lining them all up like this, you can start to get an idea of some of the ways that they could stick. And based on that, you can start to model which ones are the most sticky and folding properly. And oops, long story short, you end up with all of these different scaffolds, basically. It's very pretty, but these are the potential, folding states, basically. And then you can start to predict kind of the thermodynamics of the binding cleft in terms of, what the dissociation constants and the binding affinities would look like for these potentially different states. So like here's a good one, for example. So what this effectively means now is that if I print this out and we're going to be sticking this onto different substrates so that it won't just be one of these. You'll have like almost a bouquet of these on some other nanoscale object, basically. But these guys are going to prevent the virus from being able to stick. So they're going to block the virus from sticking to ACE2. And then the second thing that we did was create the reverse complement. So now we're going to go back to the virus here. Here's the spike, right? And here is the reverse of this. So this guy is actually based off ACE2, and it's got these two domains from ACE2 here. But in contrast to ACE2, it's much smaller. So ACE2 is basically this giant, scaffold, relatively speaking, like 600 something amino acids. This one's 50, but it just reproduces that binding pocket there. So basically, by making these two sets of binders, we can inhibit the virus from binding ACE2. We can inhibit the virus from getting coded in ACE2. And we can also mimic the immune presentation of specific epitopes that we want, basically by having the binding domains, but then also having these separate immunoepitope regions. And this is something that we can broadly use for like antiviral defense and predicting how pathogens are going to behave and how they bind and which parts you want to generate an actual immune response against. So in sum, that is kind of what we're working on, and it's the company Ligandal, we just have this very broad ability to design these sort of binders against different proteins by mimicking the existing protein and then doing thermodynamic modeling to get what we want. So, how am I doing? You guys have any questions so far? Yeah.
Hey, Andre.
Hey.
Quick question. So, first of all, the colors are a little confusing here. So the green here is just a subsection of the red, which is the binding region of the red. Is that what you're saying?
Yeah, exactly.
Okay, fair enough. How come a whole bunch of the red, which appears to be close to the complement molecule that you're showing, is not green?
Oh, it's not just proximity, it's also what kind of interaction you're having. So basically in chemistry and physics, there are different kinds of interactions. Among them, you basically have, positive charges sticking to negative charges, positive charges repulsing from positive charges, negative charges repulsing from negative charges. Hydrophobic amino acids like oil, and they like themselves, but they really hate hydrophilic water-loving ones. And the same is true for like hydrophilic water-loving amino acids liking hydrophilic water-loving amino acids, and they form hydrogen bonds. So between those, you basically have the major classes of interactions. You have hydrophobic, hydrophilic, hydrogen bonds. You have
and electrostatic
salt bridges, exactly, and the repulsory versions of this. So yeah, and then if you look at this here, for example, just to pick a random one, that guy is hydrophilic, and it's probably sticking to that guy, which is also hydrophilic.
Cool. Okay, another question. Does it really work to kind of just sort of I don't want to say blindly, but sort of in this kind of, let's just use the word blindly, it's not exactly the right word, create complementary fragments, particularly for ACE2. I mean, don't we need ACE2? You can't just disable ACE2 body-wide, because ACE2 is there for a number of reasons, right? It just happens that the virus takes advantage of that receptor. But is it really feasible to put in some drug that's going to like disable every ACE2 receptor in your body? And aren't there, by the way, already drugs that inhibit ACE2?
Yeah, well, there aren't too many ACE2 inhibitor drugs. Actually, there are plenty of ACE inhibitors, but fewer ACE2 inhibitors. And also, they're not necessarily going to bind to the right cleft. So like you have to keep in mind, this is like where ACE2 gets bound to by the virus, but I'm not actually certain that the activity of ACE2 in cleaving angiotensin into its shorter fragments is the same part as the binding region here. So I'm not even sure that the binders are going to disable the activity of ACE2.
Thank you.
And then secondly, a large part of it is that I think the virus actually absorbs the ACE2 and it forms like a shield around itself in the form of ACE2, which capacitates it from the immune system and basically hides that way. So by building something that mimics the virus, you'd be able to pull in the ACE2 and form like a shell. And then secondly, to sort of take on the opposite approach, that's also why we're making the binders that mimic the ACE2.
The spike. Yeah.
Sure. Yeah.
And so then another question. Okay, so whichever strategy one uses, let's look at the second strategy you talked about, the things which actually bind to the spike. Don't you need how can I say this? On the one hand, it would be very nice if you clogged up all the spikes and the virus just couldn't go anywhere, couldn't get into the cells. But don't you want a thing which somehow, none of this is useful against infected cells, right? Infected cells, the virus is already in there. Like you've got the cell presenting all kinds of crazy stuff.
Yeah.
How does this help with that? Or is this just designed to be more of like a antibody kind of therapy?
Oh, so yeah, I think this will have more to do with after the cell dies and it secretes a virus and it goes into the blood, it's not going to be able to get into subsequent cells. And also this binder is designed very specifically to still allow the presentation of the immunogenic portion of this.
That's not me making all that noise.
Someone's got something going on in the background there. But basically, this binder is going to still allow for antibodies to form against here. Whereas, like if you just pump someone full of ACE2, which is another approach people are taking, it's going to be like a bigger thing, and I don't think the antibodies are going to be able to fit in there and actually form the same immunoepitopic recognition. I mean, it's just hypothesis, but so I think it's a combined approach. And then the last, yeah, go ahead.
No, finish. Go ahead.
No, I was just going to say the other thing is that part of the ACE2 binders approach is going to be that we can basically look at this, look at this guy, for example. So it's got the inhibiting action here, but then it's also got these immunoepitopes. So it's going to actively train, actually here's like the full sequence, for example, but this guy's going to have this immunoepitope region, and then it's also going to have this immunoepitope region and that immunoepitope region. So basically it's going to have an antibody region, an MHC1 loading T-cell receptor responsive region, and an MHC2 T-cell receptor responsive region, which I predict are the most active ones that you actually want. So it's going to be a very tailored immune response against just like 3 immunoepitopes, in contrast to the native spike. And then there will also be a version that just has the one antibody epitope that forms a neutralizing antibody. But in contrast, like a vaccine that looks at forming the whole spike, for example, if you just print out this whole thing and you throw it in the body, normally this guy is basically pointing outward, right? And it's like, it basically is coding the virus, right? But then if you just throw the whole thing in like this, then suddenly it could be facing this way, it could be facing that way, it could be facing that way. So the antibody response is going to be broader. And also normally when this is produced in a virus, it's heavily glycosylated. So it has all of these spots along the surface that are modified with sugar domains that are actually hiding. So when you create like a recombinant vaccine or an mRNA vaccine to just produce the whole protein, suddenly you have all these parts exposed and you can start generating immune responses against it that give your body almost like a false hope. And then you get exposed to the whole pathogen and you don't necessarily have even a neutralizing antibody against this binding cleft. So that's my main concern right now with a lot of the other vaccine approaches people are taking, which is why I wanted to create kind of an all-in-one solution and really go the route of synthetic vaccines, which can be more readily scaled and just built for all these different applications.
Right. So, but if you're talking about epitopes, you know, do you actually engineer both the epitope and the MHC1 or 2 fragment? So it's like a whole tetramer kind of structure?
Yeah, exactly. So you trimer here, but you have the antibody binding epitope against the neutralizing portion of the virus.
Mm-hmm.
And then you have an MHC2 and an MHC1 epitope that are derived from deeper in here. They're actually pulled from over here, these pink parts, and they're moved up to here in such a way that doesn't mess with the binding structure to ACE2. So they're over here as opposed to the binding site.
What's the idea there, that they're just going to be more immunogenic as you present them there rather than having to go through, you know, protein cleavage and presentation, like you're just kind of pre-packaged?
Exactly, and you're going to have very tailored ones against just a select number as opposed to this random spread. And you can separate out the T-cell receptor response from the antibody response.
Mm-hmm.
It's just like a new sort of approach to making a vaccine that can be more efficacious and more precise.
Uh-huh. Have you been able to validate any of this stuff biochemically, or are you still sort of at the stage of doing simulation?
Right now it's simulation. We're having them printed out right now, and then we're going to be doing surface plasmon resonance, basically to measure the binding affinities, and we're going to be doing cryo-electron microscopy to actually validate that, you know, that they look like this and that they look like that in the bound state. So we'll be getting that back, and then assuming that looks the way that we predict it looks, we'll be proceeding to basically be doing like antibody pull down assays. So you'll basically stick these guys onto like a chip, and then you'll take convalescent serum and you'll run it over and see, does like a recovered patient from COVID-19, do their antibodies stick to these substrates or not? And assuming that's the case, we can really fast track this into humans really quickly.
Are you taking more questions?
Yeah, please just keep going.
Anyone with a question, please raise your hand or tell me in the chat, then I'll unmute you.
Yeah, keep going. Questions are right, Creon.
Creon, you have another one? Let me get you.
Great questions. Just keep them coming.
Oh, thanks. I don't know how I got muted again. Thanks. Hey, one thing I'm confused about, why are you pulling these domains from like way deep in the spike protein and using them as, why are you putting them into the vaccine? What is that, what's the point of that? I don't understand.
Yeah. So you can actually look at any protein in a virus, and you can basically map the whole sequence and figure. So there are different kinds of immune responses. There's the antibodies, as far as adaptive immunity goes. There's antibodies, and then there's T-cell receptors. So T cells are more like they float around and they basically chew up the whole protein, or it gets chewed up and presented to them, and then they basically form immune responses against these short stretches of amino acids, either 9 amino acids for something called MHC1, which is major histocompatibility complex 1, and then there's also like 13-ish for MHC2. And
but I thought that's, I mean, I'm a little rusty on this, but I thought that is when the fragments are presented on the outside of infected cells.
Exactly.
So, okay, so is the idea that by putting these purple fragments into the small peptide vaccine, that's somehow short-circuiting all that, and it's as if it was presented on the outside of an infected cell but it's
Exactly.
another. Okay.
Yeah, because it's a
Interesting.
non-native sequence too, right? So if it were a native sequence, then your body would be like, oh yeah, I've seen this before. But it's non-native. So you have like a trillion T cells, or 300 billion T cells in your body, and each of them has like, a different recognition motif for a T cell receptor that is non-native. It won't react to you. If it does, then you have autoimmunity. So it's
Okay, but I don't understand. If you make this vaccine, which is a fragment that binds to ACE2,
yeah,
in the same place that the spike protein binds to ACE2,
yeah,
but it's a very small thing, this purple thing, and
yeah.
you attach onto it some small peptide fragment that would normally be presented with MHC on an infected cell.
Yeah.
But okay, and normally that would then trigger the whole adaptive,
I see what you're asking.
response. But now it's like you're presenting it on the surface of an ACE2 receptor on a normal cell that happens to be bound to this vaccine.
Well, it's not just infected cells. It's basically, if this gets into any cells and starts getting chewed up and presented on the surface, then the dendritic cells are going to pick it up and pull it out into the lymph nodes, and the T cells are going to form an immunological synapse with it.
Oh, I see. So it's a way to generate a T cell response before you're infected, ideally.
Exactly. And you could give it to someone after they're infected too. So it'd be like, oh, now the virus has a harder time getting in, and your cells are presenting like these extra epitopes that are shared with the virus. So it's like biasing your immune response a little bit. So it's going to be like both the vaccine and kind of the antidote, and then the kind of like therapeutic. And there's like a whole spectrum of peptides that you can give depending on whether you're pre-disease, during disease, or post-disease.
Is this a new class of approach? Has this been done before with these multiple binding regions on a fragment?
No, I've never seen anyone do anything like this. So it's, yeah, I mean, I've seen recombinant vaccines. I've seen some peptide vaccines that are like 30, 40 amino acids. I've seen people take an immunogenic sequence and then latch on like something from something your body already doesn't like. But I haven't seen this sort of approach. And, keep in mind, it's not just going to be the peptide by itself. The peptide's also going to be stuck to like different bouquets of things. So it will present itself more like how a virus would look, or like multivalent substrate would look normally. So we're going to make it look foreign, basically. Your body's not going to completely love it, but it will like it more than, it's not going to be like toxic. It's just going to be something that's going to stand out. It's not quite belonging.
Is that an adjuvant, or is this something else that's
You can think of it as an adjuvant if you're doing it after you get infected, and then you can think of it as a vaccine if you do it before you're getting infected. And yeah, basically. And I mean, you can always do like adjuvant approaches, like you can stick this onto like a albumin, which is what they typically do for vaccines, or like these other things called KLH, or even like bovine serum albumin, but you don't use that for like human vaccines. Basically like things that are immunogenic, right? And then typically people are also pumping you full of aluminum. I don't think we necessarily have to do that here, but we'll test maybe 5 to 10 different approaches, even like nanotubes and stuff. Like stick it onto a nanotube and see if it's really immunogenic because of that. So yeah, right now we're just having like a bunch of the stuff made, and then we're going to have these modifications on one side of it, so we can just click it onto these various substrates and then see which ones work best for generating immune responses, for preventing the virus load from increasing, or inhibiting the virus from entering cells, or in the case of this guy, basically preventing the virus from getting coated in ACE2 and then allowing your body to clear it hopefully faster.
How do you do that, in vitro or with animals?
So right now the FDA is letting people bypass a lot of animal studies, which is interesting. You can go straight to humans. So if you can basically show that your stuff is, binding the way that you want, and even preliminarily show that, for example, one of these scaffolds can pull an antibody out from human blood, then you can pretty much go straight into humans, especially if you're going more the treatment modality and you have someone who's dying from COVID or a high-risk population. You could potentially go straight into them. And then also you could dose healthy people with small amounts of these, because they're just peptides, and see if they generate immune responses under a dose response. And if the humans do, like a 5-person clinical trial, then you can basically escalate that really quickly and try to get it out there to hundreds of thousands or millions of people and fast track. So right now the question is, can we do that in the next 6 to 8 weeks, and then use that to really accelerate the process and hopefully get a vaccine out there in the next 6 to 8 months, not like 12 to 18 months or longer, which we don't even know Moderna's vaccine is going to work. So that's what we're gunning for.
Those vaccines are going to be $1 trillion drugs, and they're going to try and stop you if they think you have something better.
Yeah, I know they already are.
How?
We'll talk about that later. I mean, I posted something on the viral exploration group. It's a little bit controversial. It's intended to be. Don't worry, I'm not losing my mind. But basically, this goes very deep, and I'm not prepared to fully talk about it yet. But Creon, you're not completely wrong.
So ideally, what would be like the kind of steps that that would come next? An idea. Wait, you said a few weeks, like 6 weeks, or what did you say? You could at least,
well, the peptide. So I had to bypass a couple of things to get my stuff done, because even like NIAID signed a contract with me, and then they didn't want to actually do the work to characterize any of my stuff. And this went straight to the White House, and a bunch of other stuff is happening. So I'm not shouting conspiracy theory here or anything. And just I know more than I should, and it's going to be okay. But long story short, we are basically having to go university paths to do some characterization. Like UCSF, UC Davis are giving us access to like the electron microscopy and also the the surface plasma resonance studies to measure the binding and all this stuff. And then at that point, we're also collaborating with France and also an undisclosed peptide synthesis group that even I don't know the name of, so that they can't fuck with us. Because they did. You guys should go check out watsonbio.com. It's very interesting. New website that appeared this week that has existed for 7 years, apparently. It's very intriguing. So, and they were offering to make this stuff for me. And then they somehow did not have the bandwidth to make it within 24 hours of my communication with the White House, which is in parallel to the IBM supercomputer group, which had also been eager to work with me, suddenly saying they don't have the bandwidth to run supercomputer work on peptide binders, which also doesn't make sense. And received both emails within like 3 hours of each other, and it's strange. So we're gonna learn more about this, but it's.
So if you need HPC time, let me know. I can connect you with people at Microsoft who are also.
That would be great.
Doing all of that. Yeah.
That would be awesome.
Yeah.
That would be really great. I basically want to do folding dynamics simulation. Well, so these are already technically folding dynamic simulations. So this is like Raptor X U Chicago. But now the whole thing is like, you basically want to take each of these chains at the ACE2 binding interface, and basically you want to, you kind of know where the starting location could be, but you want to play with the rotation and stuff. So I want to be able to take like this sequence and then basically, rotate. So maybe basically build like a little machine learning algorithm where it's like, okay, it gets rotated through this axis of freedom slightly, gets rotated through this axis of freedom slightly, because we know where the binding is approximately, and then like moved up, moved down. And then for each of those, with almost no compute time, you can generate Gibbs free energy plots. So you can basically find the minimum interfacial energy of that Gibbs free energy in terms of the delta G with almost no compute power. It's like relatively computationally lax. So you basically do this for this sequence, and then this for this sequence. And then you basically create like a gamut of like the delta G values for these 9 sequences. And even now, like looking at these 9, the way they're arranged, it's almost like a chaotic electron density cloud. So it's like my hypothesis is that actually by aligning these to like the ACE2 binding region of the virus, and then running a Gibbs free energy determination in KD, I actually am predicting, for example, based on a web server that I ran, that this has like a 5 picomolar binding affinity, which I'm sure is crazy, but I have no idea. We're going to experimentally test it. So we'll have the ability to do the feedback between those and then fine tune that approach. So that's the idea.
Cool.
Yeah. And then the other thing about this is after we validate it once, we'll be able to do this for anything. So you'll basically be able to do it for like any protein target in human biology. And at some point, even with like completely de novo peptide sequences. So You know, and I'm also interested in using quantum computing if there are accessible resources around that, because one of the things about quantum computing approaches is that they're really good at determining like free energy or minimum energy states. So basically, if you express this as a minimum energy determination equation and structure it right, then the quantum computer is automatically going to permutate towards the lowest energy state, which would be the bound state. So that's kind of one of the things I want to work on. And the other is basically creating better immunoepitope mapping techniques. They're already very good. We know how to do immunoepitope mapping. There's this service called IEDB. You can go look at it. You can type in any sequence and basically pull out the immunoepitopes as HLA compatibility, MHC binding, and all of that, and TCR1 versus TCR2. Sorry, MHC1, MHC2 for the TCR binding.
Yeah, there's actually been about 4 or 5 papers come out in the last couple of weeks who've done really thorough mapping of epitopes against MHC, all the possible epitopes of SARS-CoV-2 against MHC, all the different MHC alleles.
Exactly. And then you basically could take any new virus, any novel pathogen, and then just start pulling out those immunoepitopes and sticking them onto these substrates and building not just vaccines and building them quickly, mind you. So, you know, the next time the super virus comes out, you'd be able to be like, oh, well, here we go. And then, you know, simulation pretty much instantly, or like a day. And then the next step is printing out the peptides. And you could do that at this point also in like a day, 2 days, 3 days. So my goal over the next, you know, few years is next time something like this, hopefully doesn't, but next time something like this happens, you would basically be able to do 100 kilogram scale or greater predictive manufacturing of peptide vaccines and antidotes against viruses and just start deploying them, especially because, you know, it's not going to be like global systemic right away. Like we saw it happen in Wuhan and then it expanded. So you basically lock down an area and you immediately shut it down rather than waiting it out and having to lock it down. So I think it would be feasible to be able to build like a global biodefense network around this sort of technology.
Mm-hmm.
Any other questions? I posted in the group, but feel free to raise your hand either via the Zoom feature or in the group. All right, so I think you already mentioned to some extent, I just want to like try to pick people up, you know, in terms of like what benefit can they expect out of this, you know, when could you actually help people if things went according to plan, and so on and so forth, which it seems like currently is like not a given, but you know, if like what could people do to help you, and if it went according to plan, you know, when could this start saving lives?
Yeah, well, I mean, I think we'll be able to start testing in humans if we got all of the support that we needed from the right sources. We would be able to put this into humans within, I want to say 3 months, maybe even 2 months if we're really crazy and ambitious, right? And I think that you could readily start testing this out on 100 or 1,000 people at a time, and you don't need that much material. I mean, you could probably deal with, you know, 100 people is like a gram of material or a few grams of material. It's not a lot. And this stuff's dirt cheap too. So we're looking at cost of goods, cost of manufacturing at that scale, or at a scale of thousands of people, it becomes like $1, maybe $5 per milligram or per dose. So you're basically looking at, you know, current costs of flu vaccine or even less, and completely rapid turnaround, and being able to go straight into humans in this regulatory environment if you pull the right strings and do it right. So, you know, right now it's having access to compute resources. It's having kind of the message be out there a little bit, because you can't just be silent about it. And also, you know, I think we're about to be raising again, but right now we're not. We do have a little bit of allocation, so we're going to raise like maybe like another 250K while all this stuff is getting settled, and then it's going to be a much larger round. And then Eric Greenberg joined as my co-chairman, so he's been helping me a lot on both the kind of political spectrum of things and on the financing, and he's leading a round in a company. So that's good. So I think we're pretty set on the financial front. We can still take a little bit more cash if anyone's interested in talking about it. Just reach out to me. And and also the compute resources. And if anyone has good access to even like animal modeling sources or that sort of thing for, even working up to like BSL-4 labs or contacts at, CDC or otherwise, let's talk about it offline.
All right, thank you. So what you mentioned that now with the FDA you can kind of like shortcut a lot of the kind of like red tape that was usually required. Could you talk a little bit more about that?
Yeah, well, Moderna, their vaccine skipped animals completely.
Yeah, and they're already in clinical trial, actually, here in Seattle.
Yeah.
It started a week ago.
Yeah, they completely skipped animals for their mRNA vaccine. So.
Oh my God, that's fantastic. Maybe something, how the FDA can be reformed long term, or like, how other kind of like, I mean, you had the same in a similar, oops, I'm omitting someone else. Good. You had the, I think you had a similar, I guess, kind of like moment for hope in the longevity community,
Right.
with metformin and stuff. But like if a crisis is necessary to reform those institutions, and maybe that's a good thing. Okay. We just have Tolls. Yeah, we just have Tolls Monji saying, my friend does machine learning at Janelia Research Campus Mapping Fly Brains. I can connect you guys if you could get a contact forward.
Yeah, that'd be great. Just email me. My email is dre@ligandal.com, dre@ligandal.com. Allison has my email, of course. So.
Yes, I will also post your website on the schedule afterwards. And we have a hand raised from Mike.
While we're waiting, I just want to ask Allison, do you know how long approximately it takes for the talks to be uploaded, the recordings? Because I want Ellen Heber-Katz, who's also working on vaccines for this virus, to see this lecture.
See this? Well, she'll be delighted that she can watch this lecture probably tomorrow morning. So hi, Ellen, if you're watching this then. All right, Mike is unable to unmute. I'm going to unmute Mike now. Okay, great. Mike, your question.
Oh, hi. Yes. Looks good. I was curious, is this useful for other diseases, say like an anti-cancer vaccine?
Yeah, well, that's a little bit different, but also not. You can definitely present immunoepitopes on synthetic peptide scaffolds and then get immune responses to happen to them based on the unique epitopes that are found in the cancer cells. That's one approach. Secondly, what we previously worked on before the COVID thing was actually building these peptide binders that would target, for example, a Tcell and the known markers on a Tcell. And then basically, instead of just having a free peptide, we would build a nanoparticle. So we would basically take the end of the peptide, modify it to have a charged sequence on it, and then mix that with things like DNA, RNA, and CRISPR to form a nanoparticle. And then that nanoparticle would deliver reprogramming instructions into immune cells like T cells to basically have new markers for targeting the cancer cells or deleting things like PDL1 so that they would have basically acting like a checkpoint inhibitor. So you can go after multimodal sort of approaches here at the immunoengineering level. And that was where we had generated most of our data actually with our seed funds, was doing T-cell editing. And then when COVID came out, it was like, oh, well, I don't even need the gene delivery part. I could just build a peptide to do this thing. Granted, in like our patent filing, we also included delivery of siRNA and microRNA to turn off the genes of the virus, which is another reason Watson Bio website is kind of funny to see after disclosing all stuff to certain sources. So that's what's up. Does that make sense? I can explain that another way if you want me to clarify.
Why don't you clarify, Andre?
All right. So, okay, let's do some visual screen sharing here. So stop share. I'm going to go to T-cell receptor. All right. So let's go into Wikipedia hole here. I have a whiteboard. No, I just need this. Cool. Can you see us? All right, so normally you get your antigen presenting cells that chew things up, and then they present things on MHC1 and MHC2, and then the T cell has a T cell receptor. And so you basically have two major classes of T cells. There are actually more. You have CD8, which are like the killer ones, and then you have the T helper cells, CD4s. They're more like chill out T cells. So basically, you basically have a process of T cell receptor maturation normally, where you basically have this scrambling of the genome. So there's something called VDJ recombination, where you basically have these younger immune cells, so to speak, and they might have like 1 million DNA letters here. And they have these segments called V, sometimes they have D, and then they have J, and then they have a constant region. And basically during development, these guys get randomly mutated and shuffled around to the point that you end up with 1 V, 1 optional D, 1 J, and then the constant region. And then this is how you get immunological diversity, where you basically end up with a random scrambling of T cells that all respond to different antigens. And then the idea is that those T cell receptors, and this, keep in mind antibodies, so B cells, same exact thing, same exact process of VDJ recombination. Basically, the ones that are autoimmune get killed off during your development within your thymus, but then the ones that remain are all just like a random scrambling. When one of them binds to an antigen, it starts dividing. And basically that's how you get like ultra specific, more mature T cells and B cells against different pathogens or cancer cells. So a lot of cancers have mutagenic profiles. So they start forming like new antigens during mutation. And they have various ways of also cloaking themselves from the immune system. But one of the things that came to be was CAR T and TCR therapies, where basically you would modify an immune cell to have a new receptor on its surface, either like a synthetic biology one, which is the CAR T stuff, or a new T cell receptor. So you have to basically splice in the genetic code you want into the nucleus of that T cell by delivering things like CRISPR or viruses. And then those guys will basically be able to latch on to the tumor cells and kill them. So what we are doing with that side of things is building these delivery systems that can do this all in one step, rather than the current approaches where you have to take like the cell out of the body and then zap it with an electrical field, get your CRISPR in there, snip out some genes, and then deliver a virus to edit the genes back in. That process would cost like $270K for one person with like Novartis's therapy, this thing called Chimera. Which is, this was the first FDA-approved gene therapy. They basically did this CAR T stuff to target cancerous B cells, leukemia, lymphoma stuff, and the drug costs $500,000. So that is kind of how we want to go after cancer, but we're doing it all in one with a nanoparticle that's peptide-based that wraps up the CRISPR. You can see some of it on our website, actually. We basically have us, well, we were selling CRISPR kits basically to package up CRISPR using peptides and then do editing. And we had quite a bit of work for taking the same thing as what we do with COVID here, which would be like the gray triangle here is a binding domain. And then we would modify it to have a little charged tail and then mix it with RNA, DNA, CRISPR, you get a nanoparticle to form. And then that allows you to basically, you know, in this case, in this video, like here's a single T cell, the green stuff is CRISPR, and we delivered it with nanoparticles. And then we could do like functional gene editing to edit the T cell receptor gene. So that's one of the ways though that you can treat cancer. And I could go into that whole thing for much longer, but that's the idea essentially.
All right, any questions? Raise your hand, please, and I'll get to you. Let's see. All right, I have a question. I mean, like I've known about Ligandal that you know, for quite some time now, and I think it seems like, you know, you're like really rising to the occasion right now. But like, what are other kind of future kind of like, in an ideal case in like 10 years or so,
Yeah.
you know, what are other things that you could be applying this to? Like, is there like, you know, is there any cap to this?
Oh, I mean, in theory, this would allow you to target any cell and any receptor in the body and deliver any range of instructions, both transient and permanent in nature. So deleting, inserting, temporarily turning off, or inserting any gene you want and doing it in any cell or tissue in the body. So obviously it's a, you know, it's kind of scary. So that's why, you know, I think we're approaching it methodically. I think some of this vaccine stuff is really good. I think some of the other applications that involve gene editing need to be really carefully considered. And I think that, you know, there are certain lines that you don't want to cross when building something like this. At the same time, I see this having applications in regenerative medicine, like every rare disease that's genetic in nature. And then as we learn more about different disease states and how like maybe shifting a few genes temporarily one way or the other can ameliorate a disease state or lead to, you know, rejuvenation of an older organ, or getting your immune system to attack a specific kind of cancer, or you know, the list goes on and on and on. But you'll basically have a range of therapies where you can deliver these cures and therapies to anything ranging from cardiac tissue to neural tissue to cancerous tissue and different stem cells and things like that. So.
Oh man, then, you know, this, I mean, if it's one good thing that comes out of the crisis, that things like that, you know, have an easier way to kind of like do the good that they can do, I think that's that wouldn't be the worst.
Yeah, Allison,
right.
that was my benefit number one in my talk, was that we were going to have these great advances in immunology and virology and treatments for all sorts of diseases, just like we did with HIV, you know, which was the first renaissance and all this stuff when all this T-cell stuff was figured out.
Yeah, well, I mean, it's interesting because even in the last 2 weeks, month, the amount of resources that I've gained access to and the amount of sort of groups that have come out of the woodworks as being able to solve individual problems like around this, it's quite impressive. I think the world is really working together, and I think that you know, I'm excited to see that, because it was a lot of siloed research and everything, and people don't want to share anything, and suddenly it's like, you know, even here are my sequences, guys. Like, oh, you want to go make these? Go, you know, be my guest, please. Like, you know, that's what I'm saying. So, you know, that is kind of different from how we used to look at a lot of this stuff, where it'd be like, okay, I got to patent this, and it's going to be secret, and oh, I don't want to give this to here or there or there. And now I think it's more collaborative, because it's like, I mean, I'll be honest, I think we could easily lose 1 million people in the United States in the next 30 days, and that's conservative. So at this point, that is very real. You know, we're having, what, 10, you know, thousands. I think we hit 10,000 people dead per day.
Yeah.
It's really
scary. No, not yet. 10,000 new infections, perhaps.
You just posted it the other day.
All right. Well, I mean, I'm kind of like terribly curious as to the more conspiratorial aspects of this, but I also don't want to push you if you know, if you want to hold back with this for now. Are there any other questions?
I posted,
all right, I think
the latest numbers on death on COVID on my Twitter the other day. Let me just post up. So, sorry, 1,000 deaths per day, and it was 100 last week. And so the next 2 weeks we'll have 10,000 deaths per day, and in the next month we'll probably have 100,000 deaths per day if this keeps up. And I think there will be some sort of lockdown in that interim, but it's looking really looking crazy. So yeah.
Okay, thank you very much, Andre. Got on.
Yeah, me too. I
think
actually,
it can't happen fast enough.
Thank you.
All right, so I would say, Ravi, please get some computing going, and everyone else who I think Tulsi, connect him with your university friend. I will share your information on the schedule. Let me know if there's any other information that people should see. I will have this video done by tomorrow, and I think yeah, you've really blown people away. I've seen a lot of quite impressed and excited and curious faces here. So thank you so much for joining. What a wild modeling. And yeah, I wish you the best of luck. And I'm hoping that we may be able to have you back in a few weeks with really positive results. Let's see. All right. Thank you so much, Andre. Have a lovely night, everyone.
Bye. Bye, guys. Thanks.